Today's topic is glycine metabolism. Glycine is the smallest of all amino acids and it is nutritionally non-essential amino acid and it is glucogenic amino acid and optically inactive as there is no asymmetric carbon atom. So glycine is the smallest of all amino acid and it is nutritionally non-essential. You have studied the essential amino acid, but glycine is a non-essential amino acid and it is glucogenic and then optically inactive because it doesn't contain asymmetric carbon atom. So you can see the chemical structure of glycine where there is no asymmetric carbon atom. So it is optically inactive. Sometimes the question will come, name the optically inactive amino acid and write the important products formed from it. So then you have to know which is the amino acid. So glycine is optically inactive amino acid, which is the smallest of all amino acid and it is nutritionally non-essential. Now we will see what are the functions of glycine. Glycine is one among the common amino acids found in the protein structure. Just like any other amino acid, glycine is also one among the amino acid which is seen in the protein structure. Glycine is seen where the polypeptide bends or turns. So usually being the smallest of all amino acids, it is usually seen where the polypeptide bends or turns. That is, it can fit into the bends or turns. Collagen contains a very high content of the glycine. That is, 30% of amino acids of collagen is made up of glycine. So 1/3 of amino acid is made up of glycine in collagen. So collagen contains 30% of glycine. Second important function is glycine participate in one carbon metabolism. Okay, so the glycine is one among the commonest amino acid found in the protein structure, mainly collagen. Collagen contains about 30% of glycine because it is the smallest amino acid. It can fit into the polypeptide bends or turns. Then it is participating in one carbon metabolism. Now, glycine also involved in the synthesis of many specialized products. So, one of the important function is it is involved in the production of various biologically important products like purine bases. You know what are the purine bases? Adenine and guanine. Then glutathione. It is a tripeptide. Then creatine. You know what is creatine? It is present in the muscle. Then formation of bile salts, that is from the bile acid bile salts are formed, and then for the formation of heme is required for the synthesis of hemoglobin. Then serine, another amino acid, serine can be formed from glycine, and hippuric acid is produced from glycine, and then glycine is acting as a neurotransmitter, and also glyoxylic acid is produced from the glycine, and finally of course the collagen we have discussed already. So these are the important products formed from glycine, that is purine bases, glutathione, creatine, bile salts, heme. Actually the first step in the heme synthesis utilizes glycine. Then another amino acid, serine, is formed from glycine. Then hippuric acid, it is a conjugated substance formed by the glycine with benzoic acid forming the hippuric acid, and then it is acting as a neurotransmitter. Then glycine produces glyoxylic acid during the metabolism, and it is 30% of amino acid in the collagen is formed from glycine. So the usual question for you is the important specialized products formed from glycine. So you have to study some of the products and how they are formed in the body. So in this picture or in the flow chart also you can see it is producing protein. Serine is formed in oxalate, that is an excretory product. Then glucose, it is a glucogenic amino acid, so glucose is produced from glycine, and also just like any other amino acid, the final product is ammonia, which can be converted to urea. Then another product is formate, that will go to the 1-carbon metabolism. Then purine, whole of the glycine is utilized for the purine base formation. Then glutathione, a tripeptide, and conjugation, that is bile acid, is formed by the conjugation with glycine. Bile acid is conjugated and produces bile salts. And then the heme synthesis, for heme synthesis, first step of heme synthesis requires glycine. Then for the synthesis of creatine, which is present plenty in the skeletal muscle. So these are the overall picture where you can get an idea about the glycine metabolism. Okay. Now a few words about how they are synthesized. Glycine is synthesized in our body. We have already discussed that it is a non-essential amino acid, so it has to be synthesized in the body. So first, glycine is formed from serine amino acid. So it is producing serine. Also serine produces glycine. Serine with the help of 1-carbon transfer by the tetrahydrofolic acid by the enzyme serine hydroxymethyl transferase, that beta carbon is given to the for 1-carbon pole, and the alpha carbon is given to form the glycine. Then threonine, with the help of the enzyme threonine aldolase, threonine is forming glycine. Threonine is producing glycine. Then carbon dioxide and ammonia combines with the help of the enzyme glycine synthase to form the glycine. So from carbon dioxide and ammonia, utilizing the glycine synthase enzyme, you can produce glycine. Then from the glyoxylate, by the action of aminotransferase, that is transamination with pyruvate or alanine, you can get the glycine. And if it is alanine, pyruvate is formed. If it is alpha ketoglutarate, glutamate is formed, and the glycine is formed. So in this flow chart you can see all these. From serine, it is producing glycine by the enzyme serine hydroxymethyl transferase, where the tetrahydrofolic acid will take the beta carbon of the serine and forming N5N10 methylene tetrahydrofolic acid. So serine alpha carbon is producing the glycine. Serine hydroxymethyl transferase is the enzyme. Next is from the threonine amino acid, by the action of threonine aldolase enzyme, where acetaldehyde is removed and the glycine is formed. So this is the second method by which you can get glycine, that is from the threonine amino acid. And the third one is from the carbon dioxide and ammonia. It is forming glycine by the enzyme glycine synthase. Here again NADH plus H is utilized, and N5N10 methylene tetrahydrofolic acid is utilized to form the NAD and the tetrahydrofolic acid and glycine. So here carbon dioxide plus ammonia, with the help of glycine synthase enzyme, glycine is formed. Then the fourth method is from glyoxylate. Actually glyoxylate is a product of glycine metabolism, and glyoxylate is on transamination. You have studied transamination reaction in urea cycle, right? So transamination with alanine, glyoxylate with alanine produces glycine plus pyruvate, or if glyoxylate plus glutamate with produces alpha ketoglutarate and glycine, or glyoxylate plus serine produces hydroxypyruvate and glycine. So for all these type of reaction, glyoxylate aminotransferase, that is a type of transamination reaction. So by the transamination of glyoxylate, you can get glycine back. So these are the 4 steps by which you can synthesize glycine in our body. Now how the catabolism occurs, just like any other amino acid synthesis, there is any catabolism also. So the glycine, we have noted that the glycine is produced from the ammonia and carbon dioxide by the glycine synthase enzyme. The same enzyme, it is a reversible reaction. Glycine can be converted to ammonia and carbon dioxide by the reversal of glycine synthase, that is glycine cleavage system. Same enzyme, that is when it is converting glycine to ammonia and carbon dioxide, then it is called the glycine cleavage system, where NAD and tetrahydrofolic acid is utilized, and NADH plus H and methylene tetrahydrofolate is produced. Finally, ammonia and carbon dioxide is produced. So this is the major pathway of catabolism of glycine. Next is this glycine can be converted to serine, that is a reversal of the serine hydroxymethyl transferase enzyme, and serine can be converted to pyruvate by serine dehydratase. So we have told that it is a glucogenic amino acid, right? So how it is occurring? Serine on action with serine dehydratase, removal of ammonia and water produces pyruvate. So ultimately the glycine is converted to pyruvate, a glucogenic substrate. Then another reaction is glycine on transaminase reaction producing glyoxylate. That is again we have noted in the synthesis of glycine. Glyoxylate on transamination with glutamate produces alpha ketoglutarate and glycine. So the reversal, glycine plus alpha ketoglutarate will produces glyoxylate and glutamate by the transaminase enzyme, and this glyoxylate have got a pathway, that is on oxidation it will produces oxalate, which is excreted through urine. So finally it can be excreted through urine also, and also glycine can produce ammonia here in this reaction, that is serine dehydratase reaction. Ammonia is produced, which can also be converted to urea and excreted. So glycine cleavage pathway is the major catabolic pathway of glycine. That is, it is a multi-enzyme complex where glycine decarboxylase, methyl transferase, then methylene tetrahydrofolate synthase and lipomer dehydrogenase are the enzymes together acting as a glycine cleavage system, and the reversal is called the glycine synthase. So remember, the major catabolic pathway of glycine or the degradation of glycine is through the glycine cleavage system, that is a multi-enzyme complex containing glycine decarboxylase, methyl transferase, methylene tetrahydrofolate synthase, lipomer dehydrogenase, and all this actually this is the reversal of glycine synthase, where the ammonia and carbon dioxide used to form the glycine. Okay. So this is the reaction where the glycine is converted to ammonia and carbon dioxide, that is carbon dioxide and ammonia is released, and from the carbon dioxide and ammonia glycine can be formed by the glycine cleavage system. Forward is the cleavage system, and then ammonia plus carbon dioxide can form the glycine also. Now we will see what are the important or the specialized products formed from glycine individually. So we already studied that it is used for the synthesis of protein, collagen, then conjugation reaction with benzoic acid to form hippuric acid, formation of bile salts, glutathione, heme synthesis, purine, purine, that is then one carbon metabolism, and also for the creatine, and also it is used for serine, synthesis of serine, and also serine can be converted to pyruvate, and for pyruvate it can be used for the synthesis of glucose through the gluconeogenesis. That is why it is called the glycine is a glucogenic amino acid. Is it clear for you? So all these products are formed from glycine. So usual question is, what are the important substance derived from glycine? Describe their steps of formation of any two. Glycine is a constituent of protein that we have mentioned already, and it is participating in one carbon metabolism, and glycine involved in the synthesis of many specialized products. First is we are going to study the formation of purine ring. So the entire glycine is utilized for the formation of purine, that is carbon number 4, carbon number 5 and nitrogen 7 of the purine is produced from the glycine. The whole of the glycine is utilized. See, numbering of the purine ring is like this: 1, 2, 3, 4, 5, 6, N7, C8 and N9. So here carbon number 4, carbon number 5 and the nitrogen 7 is from the glycine. So you can mark these C4, C5 and N7 and can write there it is from the glycine. So the whole of the glycine is utilized for the formation of purine base. Second one is biosynthesis of creatine. So you know what is creatine. You have studied the organic constituent of urine. One of the important organic constituent of the urine is creatinine, and it is the anhydride of creatine. So creatinine is formed from the creatine, which is present in the muscle as creatine phosphate. Creatine is present mainly in the muscle, that is 95% is present in the muscle and rest is present in the brain or heart. Creatine is constitute about 0.5% of the total muscle weight. There are 3 amino acids which is required for the formation of this creatine, that is arginine, glycine and methionine. So 3 amino acids used for the synthesis of creatine are glycine, arginine and methionine. So we will see the steps of synthesis. That is, first the arginine, guanidine group of arginine combines with the glycine to form the guanidoacetate. That is taking place in the kidney, and it is the first step we are synthesizing guanidoacetate. The reaction is taking place in the kidney. In the next step, methyl group from the S-adenosyl methionine, that is SAM, S-adenosyl methionine, donate methyl group to the guanidoacetate by the enzyme guanidoacetate methyl transferase to form the creatine, which is taking place in the liver. So the first reaction is taking place only in the kidney, not in the liver. The second reaction, that is the transference of methyl group to the guanidoacetate to form the creatine, is taking place in the liver. So now we have synthesized the creatine, that is finally synthesized in the liver. So what happens to the creatine? In the creatine later on is acted upon by creatine kinase enzyme. We have studied creatine kinase in isoenzymes or clinical enzymology, where there are isoenzymes of creatine, that is creatine kinase, MB, etc. So this creatine kinase is present mainly in the muscle, and that in the muscle it will combines with ATP to form ADP and the creatine phosphate. So the creatine is converted to creatine phosphate. The phosphate group is donated by the ATP in presence of the enzyme creatine kinase. Now this is a temporary storage form of energy in our muscles. When the muscle contracts, usually the ATP which is present in the muscle will hydrolyze and utilize the energy. So again, if you need energy, that will be given by the creatine phosphate by the action of creatine kinase. That creatine phosphate is mobilizing the high energy phosphate bond to the ADP to form again the ATP. So that reaction is called the Lohmann's reaction. So here what happens to the creatine in the muscle? You can see creatine is first acted upon by creatine kinase to form the creatine phosphate, and then creatine phosphate is hydrolyzed to anhydride of creatine or creatine phosphate is the creatinine. So here creatine on removal of water, it is cyclized to form the creatinine. Similarly, here also creatine phosphate by the removal of inorganic phosphate, it is converted to cyclized form of the spontaneous cyclization to form the creatinine, which is excreted through urine. So the end product of creatine metabolism is creatinine. That is what you are estimating in the urine. Urine contains creatine. So the creatine usually is a normal organic constituent of the urine. So in the muscle, after creatine phosphate is converted to creatinine and excreted. So the whole pathway is glycine. First combines with arginine, guanidinoacetate is taken and ornithine is released by the transaminase enzyme, forms the guanidinoacetate. Then methyl group is donated by S-adenosyl methionine to form the S-adenosyl homocysteine, and the enzyme is methyl transferase, and guanidinoacetate is converted to creatine. Now the creatine in the muscle, second reaction is taking place in the liver, and the first reaction is taking place in the kidney. Then creatine acted upon by ATP in presence of the enzyme creatine kinase, which is taking place in the muscle to form the creatine phosphate, or it is also known as phosphagen, and then finally it is hydrolyzed and cyclized to form the creatinine, and it is excreted through urine. Now, what are the clinical importance of creatine and creatinine? Blood level of both are constant as long as the muscle mass is not affected. So it is present, we have already mentioned that 95% is present in the muscle. So its level is almost constant till the muscle mass is constant. The serum level is creatine is 0.2 to 0.6 milligram per deciliter. Creatinine in the blood is 0.7 to 1.4 milligram per deciliter. So creatine, we are estimating usually the creatinine level in the blood as a routine renal function test. Then urine creatine is 50 milligram per day, and the creatinine in the urine is total 1 to 2 grams per day. So this is 24 hours value. Usually urine we will express per day, because 24-hour urine collection, we are after collecting 24 hours urine, we are estimating the either creatine or creatinine. So creatinine on 24 hour is 1 to 2 grams per day. Serum creatinine level is the 0.7 to 1.4 milligram per deciliter, which is the important renal function test. So creatinine is an anhydride of creatine and formed by spontaneous cyclization of creatine, that we have already seen in the previous slide. Now what are the clinical importance? Estimation of serum creatinine is used as a diagnostic test to assess the kidney function. It's an important renal function test. In muscular dystrophies, serum creatinine level and the urine creatinine levels are increased. Then increased output of creatinine, that is creatinuria, is found in muscular dystrophy, diabetes mellitus, hyperthyroidism, and starvation. In all these conditions, increased output of creatinine is seen in the urine. Now the next important product formed from the glycine is for the synthesis of heme. Glycine condenses with succinyl coenzyme to form delta aminolevulinic acid. That is the first step and the rate limiting step in the heme synthesis. Okay, so that is the first step. So if the glycine is not there, this first step cannot be performed and the heme synthesis will be affected. So glycine is required for the synthesis of heme. Glycine condenses with succinyl coenzyme. Where is succinyl coenzyme we are getting? That is from the citric acid cycle. It is an intermediate of citric acid cycle. So glycine combines with the succinyl coenzyme to form the delta aminolevulinic acid or ALA by the ALA synthase enzyme. Delta aminolevulinic acid serves as the precursor for the heme synthesis. So you can see the reaction here, succinyl coenzyme plus glycine. ALA synthase is the enzyme forming delta aminolevulinic acid. So this is the rate limiting step in the heme synthesis. Next is glycine acting as a conjugating agent. How it is acting as a conjugating agent? So bile acids are the bile acids usually conjugate with glycine. You know how bile acid is formed in our body? That is from the cholesterol metabolism. Bile acids are formed and it is conjugated with glycine to form glycocolate or glycosinodeoxycholic acid. So cholic acid plus glycine forms the glycocholic acid. Then cholic acid plus glycine form glycosinodeoxycholic acid. So these are the bile salt, primary bile acid. Another detoxification reaction is detoxification of benzoic acid to hippuric acid. We have already mentioned in the product form hippuric acid is produced from glycine. How it is occurring? The benzoic acid is used as food additives for storage of various foodstuffs. So when it is present in excess quantity in the body, it has to be detoxified. So this detoxification is made by combining the benzoic acid with glycine to form the hippuric acid. So you can see the reaction here. Benzoic acid combines with the glycine to form the hippuric acid, and it is made water soluble and easily excretable form. So the harmful benzoic acid is detoxified to less harmful or non-toxic substance hippuric acid, which is easily excretable through urine. Another important product is synthesis of glutathione. Glutathione is a tripeptide. It is synthesized from 3 amino acids, that is glutamic acid, cysteine, and glycine. So it is a tripeptide having 3 amino acids, glutamic acid, cysteine, and the glycine. So the synthesis is, you can write the synthesis also by stepwise. Glutamate plus cysteine forms the gamma glutamyl cysteine by the gamma glutamyl cysteine synthase utilizing ATP, and ADP and PI is formed. Now the gamma glutamyl cysteine is formed. Then the glycine is acted upon this and utilizing one more ATP with the help of the enzyme glutathione synthase forms the glutathione, which is the gamma glutamyl cysteine glycine. Okay, so the chemical name of glutathione is gamma glutamyl cysteine glycine. It is synthesized from glutamic acid, cysteine, and glycine. In the first step, glutamic acid and cysteine forms the gamma glutamyl cysteine by the gamma glutamyl cysteine synthase. Then one more one more molecule of ATP is used, and glycine is combining with the gamma glutamyl cysteine by the enzyme glutathione synthase. Enzyme is glutathione synthase to form the glutathione. Now, so there are some important functions for glutathione. Just to know what is that, for the maintenance of RBC membrane integrity and amino acid transport, you might have studied Meester cycle into the cell. When amino acid is taken, glutathione is used. Then for the preservation of active site of the enzyme, that is SH group containing enzyme, the active site is preserved and protected by the glutathione. Then involved in the phase 3 conjugation reactions by in detoxification. You know what is detoxification? That is, toxic substance in the body is made non-toxic and easily excretable water-soluble substance. That is the process known as the detoxification. So various methods are there for detoxification, out of which the third there are three methods. First, phase 1, phase 2 and phase 3. So the phase 3 detoxification is through the with the help of glutathione. So it is involved in the phase 3 conjugation reaction. So glutathione is involved in the phase 3 conjugation reaction in the detoxification of various substances. Then glutathione is also have a coenzyme role. Usually the glutathione, reduced glutathione is written as GSH and oxidase is GSSG. So this is reduced glutathione and it is used as it has got a coenzyme role also. Now another important function is glycine acting as a neurotransmitter. So the glycine is seen in the brainstem and the spinal cord, and it opens the chloride specific channels in the brain. So what happens? In moderate amount, glycine inhibits the neuronal traffic, but in high level it causes overexcitation. So it has got both action. If it is of moderate quantity, glycine will inhibit the neuronal traffic and inhibit the it is acting as an inhibitor. But if the level is very high, if the level of glycine is very high, then it causes overexcitation and fits and all will come. Now 2 or 3 important disorders are associated with the glycine metabolism. Just namesake, you have to know what is metabolic disorders of glycine. One is glycinuria. It is a rare disorder. Very high amount of glycine is excreted in urine. Glycinuria, in the name itself, you can get that it is in the excretion of glycine more in the urine. Its main defect is the renal reabsorption of glycine, and it is characterized by increased tendency for the formation of oxalate renal stones. So excretory product of glycine is we have studied oxalate. So that will be excreted through the large quantity when it is going through the renal tract, and it will be producing stones, renal stones. Then second disorder is non-ketotic hyperglycinemia. Glycinemia means glycine level will be high in the blood. So glycine level is increased in the blood, urine and the CSF, and defect is in the glycine cleavage system. What is glycine cleavage system? It is the catabolic enzyme, isn't it? Glycine cleavage multi-enzyme system is involved in the catabolism of glycine to carbon dioxide and ammonia. So if there is any defect, the level of glycine in the blood, urine and CSF will be increased. Several severe mental retardation and seizures are seen. When the level of the glycine is high, there will be a neural overexcitation, so that will be producing seizures and also mental retardation. For this, treatment is intake of diet free of glycine. So restrict the diet and completely diet free of glycine should be given. Then third one most important is the primary hyperoxaluria. That is, it is characterized by an increased urinary oxalate excretion. Here the oxalate excretion is very much increased. That is, usually excretion is up to 50 milligrams per day, but here up to 600 milligrams per day it is seen in the urine. Oxalosis also is observed. It's present in the level of oxalate will be more in the blood also. Oxaluria is due to the increased production of oxalate, and it is due to the failure to catabolize glyoxylate. It is a defect in the glycine transaminase and defect in the protein targeting. So what is the meaning of protein targeting? Once any protein, once it is synthesized, it has to be transported to the corresponding site, and if it reaches the corresponding site, then only it can be utilized. So that process is called the protein targeting. So in this metabolic disorder, there is a protein targeting defect. Actually, this enzyme glycine transaminase should be transported to peroxisome. Its main action is taking place in the peroxisomes of the cell, but here due to the defect it is transported, this protein enzyme is transported, glyoxylate aminotransferase is transported to mitochondria of the cell, so it is not active. Once it reaches the mitochondria, it is inactive, it is not active, so it will be remaining as such, and so the glyoxylate is not able to convert to back to glycine. Instead it is producing more of the oxalate, getting oxidized and producing more of oxalate. That is the cause for oxaluria and oxalosis in the body. Okay, that much you have to remember. It is a protein targeting defect where the glycine transaminase is wrongly uh, reached into the mitochondria instead of peroxisome. If it is in the peroxisome, it will act regularly and it will produce glycine, and there is no excess production of oxalate. But it is taken to the mitochondria where it is inactive, it cannot function, so it will be producing oxalate, and instead of producing glycine, it is converted, it is oxidized to oxalate and producing oxaluria. Then there is one more type 2 primary hyperoxaluria. It is a milder condition due to the defect in the activity of cytoplasmic glyoxylate oxidase. So another enzyme called glyoxylate oxidase, that is defective. So in this case only urolithiasis, stones are seen in the urinary tract. No other complication. It is a milder condition and also rare condition. So the general principle for the management of hyperoxaluria are increased water intake to increase the oxalate excretion. So usually the oxalates are seen deposited in the urinary tract, isn't it? So we have to remove it by drinking lot of water so that it will get excreted through urine. Then to minimize the dietary intake of oxalate. Oxalates are seen in plenty in the vegetable diet, that is fibers and all contains plenty of oxalate. So restrict that type of diet. Restrict the intake of leafy vegetables, beetroot, tea and cocoa. That is, these are the things which contains plenty of oxalates. So restrict these type of diets and also drink plenty of water so that whatever oxalate is produced will be easily washed off through the urine. So this is about the glycine. So for you, for the examination point of view, the main question is what are the important substance or specialized products formed from glycine, and sometimes they may ask any write the pathway of any two or any three, like that. So you have to study important products which are formed from the glycine, like here, purine base, where the C4, C5, and 7 is produced from the glycine. Then you can write heme synthesis first step, that is glycine plus succinyl coenzyme A forms the delta aminoalanic acid, which is the first step in the heme synthesis. And again you can write the pathway of creatine synthesis, that is arginine and guanodoacetate plus glycine form the guanodoacetate, then methyl transferase produces finally methyl guanodoacetate, that is the uh, creatine. Then you can also write glutathione, gamma glutamyl cysteine glycine, that is produced from glutamic acid, cysteine, and also glycine together with the help of two enzymes, and also ATPs. Two ATPs are required for the formation of glutathione. These are the important products. So if you are asked to write the product, you better write all these four, that is heme, purine base, creatine synthesis, and glutathione synthesis. If you are asked to write the function, you can write about the benzoic acid deconjugation reaction, neurotransmitter function, etc. Okay, so I hope you have understood what are the important products formed from glycine, how glycine is formed in our body, that is four mechanism, one from serine, one from threonine, one from carbon dioxide and ammonia, and one from glyoxylate. Then the catabolism, how it is degraded, that is glycine is converted to ammonia and carbon dioxide by the reversal of glycine synthase, that is the enzyme is glycine cleavage multi-enzyme complex. Okay.